Иначе, если вы так будете отправлять с тещей, она не будет воспитана тещей.
Я уже заметил. Алло, Наталья Викторовна. Здравствуйте.
Все, я могу начинать, да?
Ну, пока что здесь ожидаем студентов.
А у них есть где-то ссылки на это, когда они уже узнают, как заходить, куда?
Да,
Звук, поняла.
все. И можно листать презентацию и читать лекцию.
Ага, хорошо, спасибо. Но пока никого нету у меня.
Ну, пока
никто
никого
мне
нет.
не зашел,
Один
да?
заходит, выходит, заходит, выходит.
Здесь? Что-то я не вижу
Ну,
никого.
здесь он не пишет, но там по статистике я смотрю, там он заходит и
А
выходит.
где у нас статистика?
А я свернул.
А, это свернуть, если,
Да.
да? Ну, так они заходят, выходят, число зрителей один.
Да. Но он еще чуть подвисает, их может быть до 5, он пока отобразит один.
Ага,
Там,
понятно.
я заметил
Ну,
тоже.
Ладно, буду начинать. Все, звук у меня есть, да?
Все есть, все работает, да.
Okay, my dears, is this second or first division, if I'm not mistaken? So, guys, start to collect together here to listen to the lecture. So, we start right now, and if I'm not mistaken, this is the first division. So, within this term, I will be reading both immunology and microbiology. We will be alternating. Guys, chiefs, please collect your groups. I can see that only few students are participating in a lecture. Guys, collect together. So I think around 4 students are there. Is it true? So now, this is a lecture in microbiology, and today the topic of today's lecture is the very dangerous bacterial infections, very dangerous bacteria, causative agents of dangerous infections. And those agents that we will discuss today can be used, as you see here, as biological weapon. So, and we have many lists of these biological weapon organisms. So, but then out of all, in the short list we have Yersinia pestis, Bacillus anthracis, Vibrio cholera, and as an example of some other groups of bacteria that can be recognized as potential biological weapon, we may have Borrelia recurrentis that may cause louse-borne epidemic relapsing fever, LBRF briefly affiliation, and Vibrio and Rickettsia prowazekii that may cause epidemic typhus, historically important disease, and Vibrio cholera. Guys, have you collected together? Okay, so we used to say that these microbes may cause biohazard of disease, or they may be a biological hazard or biohazard. What is it exactly? So biohazard is a biological substance that possesses a threat to the health of living organisms, mainly humans. This could include a sample of a microorganism, virus, or microbiotoxins that can adversely affect human health. A biohazard could also be a substance harmful not to humans only, but also to animals. And you can see here the sign of biohazard disease. So this is the sign that you can see on the doors of some clinical labs, medical labs. So, and usually we measure the dangers of the microbe according to the biohazard level, that more commonly may refer to as biosafety level or biological safety level, briefly biosafety level. These levels are the classification of safety precautions necessary to be applied in the clinical microbiology lab, depending on specific pathogens handled when performing laboratory procedures, diagnosis, culture, or whatever. And the complexity of each biohazard or biosafety level aligns with many different properties of the microbes. For example, infectivity, severity of the disease they cause, the ability of the microbes to spread, and so the more freely they spread, the more lower or the more strict safety precautions should be undertaken to deal with this microbe. So many different features of the microbes can be considered when we identify the biohazardous level. And of course, the nature of laboratory work is also important. And I would like to come back to this list, and according to the CDC control, the Center for Disease Control, the most harmful microbes are placed into the biosafety levels 3 and 4. For example, in biosafety level 3, we have out of here Bacillus anthracis, Vibrio cholera, Borrelia recurrentis, Rickettsia, Yersinia pestis. And then if you go deeper into biosafety level 4, we have there some mainly viral infections. Ebola, Marburg, some virus, hemorrhagic, the viruses that may cause hemorrhagic fevers or spotted fevers or whatever. So mainly bacteria are located in the biosafety level 3. By the way, HIV is also enter the biosafety level 3, not the highest. Okay, so we go ahead with the first microbe in the list, that is Yersinia pestis, the causative agent of plague. This is really a very serious infectious disease, and quite often it can be lethal or deadly. So, according to the different forms of plague that we have, most common are bubonic plague and pneumonic plague. So, and for bubonic plague, the mortality rate, if not treated, 50%. And you can see here, then later on you will see some other pictures. And the second form, that is pneumonic plague, that is 100% lethality. So, and these bacteria may circulate in nature with no any connection with the human. So this is not human disease. This is animal disease that is called zoonotic disease. So it can be transmitted occasionally from animals to humans. So that's why we have a natural source of this disease. And the natural source is the infected rodents, mainly small rodents. But more than 200 different species of rodents and some other types of animals can be infected. So the question is how this disease is transmitted from animals to humans. So usually the transmission occurs through vector. So the bubonic plague, so-called, is transmitted from animal to animal or from animal to human by the bites of the fleas. That is an example of the flea that you studied on biology department, medical biology department. So let's take a look at the Yersinia itself. So before it was placed together in the same family Pastorella, right? Because Yersinia, Francisella, Pastorella are these three genera that have several common features. They have animals and as their natural hosts. Then they can cause diseases in humans, and they are pleomorphic, gram-negative rods. You see pleomorphic, so they look like rods, but some of them can be like cocobacilli, larger or smaller, curved a little bit. That's what we call pleomorphic. So then, one more feature, they can exhibit so-called bipolar staining. And later on we will see the ends due to the staining are darker with lighter middle. And that is called bipolar staining because the poles are stained better than the middle. So they also can be characterized by ability or disability to produce particular enzymes. So usually they are catalase positive and oxidase negative. And regarding the oxygen requirements, they are facultative anaerobic or microaerophilic bacteria. So first few words about the disease they cause. I think you've heard about so-called plague. And within the human history or history of the mankind, we may distinguish several epidemics or pandemics. And the first very well described pandemic occurred in the 6th century AD, Anno Domini in Latin, and killed 100 million people in 50 years. The second major pandemic that's supposed to be called and used to be called Black Death originated in Asia in 14th century and spread to the Near Eastern Europe, and it occurs within several centuries, 14th, 15th and 16th. In Europe alone, 1/4 of population died of this disease. That was this one, second one, this most devastating epidemic within the history of the mankind. And the third pandemic originated in Burma in 1890s, spread to Chinese, and from there it spread to other continents, and how it spreads through the rat-infested ships. And this spreading led to the establishment of these organisms in wild rodents in many, many countries. So now you will see later on the map of the distribution of Yersinia pestis. And you can see here the picture of the very well-known artist Peter Bruegel that shows the epidemic of plague in 16th century that is called the Triumph of Death. And you can see here that any humans could be infected. You can see the king dying on the street, that is here, right? And you can see dogs eating the corpses on the streets, and the corpses are there laying in the huge heaps. And you can see monks in the carriage that are performing the burning over the corpses, and really it demonstrated inescapability of any people from this disease, and really the hard times for the Europe when they have this epidemic. So now, how about plague today? You may think that that's just a history. No. But then, of course, the incidence of this disease is very low. But from 210 to 215, there were 3,200 cases reported worldwide, including a bit more than 500 deaths according to WHO. And you can see here the number of reported human plague cases in pink cases, and in dark brown deaths. And according to this picture, we can see that the main hot spot in the world is Madagascar island. You can see that the highest proportion of the cases occurred there. Then you see a hotspot in Congo. Then the United States, but then the incidence is not so high there. United States, Peru, then Tanzania, a bit in China, Mongolia, very few in Russia. And in red color you can see the potential natural focuses for plague. What does it mean, natural focuses? It means that the plague occurs there in nature, in wild animals. And not so far ago, several cases emerged in Mongolia, and in Mongolia, this is China province, And 2 deaths occurred this year within July and August, and the hunting and eating of marmots has been prohibited because these are, you can see here, the small boy, they died of the plague not so far ago, a month ago, because he had been hunting these small animals, and this is a delicious, they are delicious in Mongolia and China, so that's why from time to time they may get this infection. So it won't disappear today. Now a few words about the organism itself. You can see here what does it mean bipolar staining. On here you can see it appear as safety pin. This is a gram-negative rod, very small, non-motile, non-sporing, and bipolar staining can be seen due to the methylene blue staining or some other staining. Yersinia pestis is sensitive to heat, sunlight, drying, chemical disinfectants, but it takes up to 15 minutes to kill it by phenol, for example. But then the thing is that it is very resistant in the environmental conditions. So it likes cold, moist environment, and it can survive there for very long years, and it remains viable for 1 year even in the frozen infected fleas if you collect them and put in a freezer. And it may also remains viable for half of the year in the soil of rodent burrows. So that's why, as you understand, it is impossible to get rid of it from nature, I mean. So it will occur always as a natural disease. So how about the cultural biochemical properties? They have quite unusual properties. First, they are not exacting in their nutritional requirements and may grow on MPA media very well. Then, unusually, they may grow over a very wide range of pH, from 5 to up almost to 10, 9.6. And it also may grow at the very wide range of temperature from 2 to 45. Can you imagine? So, and the fastest growth occurs at 30 degrees Celsius. So usually we use McConkey agar as a selective medium to grow it. And it is lactose negative, so it forms colorless colonies on McConkey agar. So it likes blood agar, and its colonies are dark brown because they absorb amine very easy, and the colonies are very small, transparent, like drops of water there. You can see an example. So, and if we have a virulent strain, then it will produce the opaque colonies. The important characteristic of the microbe is the virulence factor that it may produce to cause harm to the human body. And of course, because it is gram-negative bacteria, it has lipopolysaccharide. Then, on the surface of the microbe, there are some protein molecules. It is called fraction 1 molecules. And this is antiphagocytic envelope protein. And what is unusual? It is what is usual is it is heat labile. So, interestingly, that a lot of virulence factor of Yersinia pestis, they encoded by the variety of plasmids. So, F1 is encoded by P fraction plasmid. Then the second very important, the third virulence factor is V and W antigens that are encoded by one more plasmid, PCAD plasmid, that it is a plasminogen activator gene. So these antigens may degrade complement components, and if it is degraded, so it may inhibit phagocytosis. Coagulase can block preventriculus of the flea. Coagulase, this is an enzyme that is not produced in the human body, but it is produced in the flea body. So at 28 degrees Celsius, but not at 35 degrees Celsius. It is aiding transmission of the infection from one susceptible, from infected organism to the susceptible organism. So, and it is also encoded by P. pestis plasmid. Then they may produce fibrinolysin that is encoded by one more P. pestis plasmid. And it degrades fibrin clot and permit Yersinia to spread readily in the body from the blood clot. So that's why it may cause dissemination. We go ahead with the virulence factors. So the Yersinia pestis has a large family of the proteins, altogether around 11 proteins that are called outer membrane proteins, or briefly YOP proteins. They are components, and you can see here, of the type 3 secretion system. What is it? It is a very effective tool or molecular syringe that allow the bacterium, this is a part of bacterium, so, and this is a part of the human cell. This is a cell membrane that allow bacteria to inject the outer membrane proteins from the bacterial cytoplasm to the human cell cytoplasm. And this is the simplified scheme of the mechanism. And here you can see scanning microphotograph, and where you see really that this pili, third type pili, exist and have needle structure and important in transportation of the toxic products from the bacterial cell into the human cell. Yersinia YOP proteins, they are toxic, cytotoxic proteins. They may inhibit the migration and engulfment of phagocytosis and inhibit platelets aggregation, so it may interfere with the survival of bacteria, so they increase survival of bacteria in the human body. Some more factors, for example, pesticid or bactericid of Yersinia pestis inhibits E. coli, Yersinia enterocolytica, and Yersinia pseudotuberculosis. Piscicins also is a product of the plasmid gene expression, and this also can be considered as one of the virulence factors. So, except for the outer membrane proteins, so it may also have so-called YAD genes, Y-A-D. So these are proteins for adhesion, because adhesion is also a very important step in the infection. Yersinia pestis is toxigenic. Except for endotoxin, it may produce exotoxin, which is called usually murine toxin, because it leads to death of the small rodents in very low concentration. It is also called fraction 2 antigen. This is a thermolabile protein that may be toxoided, so it can be used in the treatment or prevention. And here you can see on the scheme the virulence factors. So, and here you can see YAD genes, right? These genes that encode for adhesins and invasins here, right? So, because through these adhesins, bacteria may bind to collagen, fibrinolactin, to the mucus, so that's why to many different types of human tissues, to the hosts. Then we mentioned that other factors inhibit the immune response of the host. So, for example, plasminogen activator. And also we can see here the type 3 secretion system proteins out of your protein that may transduce some signals from the bacterial cell directly into the human cell to activate the pathways that may lead to the protein. So your protein is a protein kinase, for example, that induces macrophage apoptosis, so it may lead to the death of the macrophages. And also they could not stop the multiplication of bacteria. Clinical forms of plague. So let me see, do we have any other? Okay, this is strange. I wonder why no people are coming for our lecture. Those guys, if you hear me, please call to your group mates. Let them to connect to our lecture and listen. Okay, clinical forms of plague. We have 3: bubonic plague, pneumonic plague, and septicemic plague. So, of course, the most common one is bubonic plague, and that is most mild infection. But asymptomatic infection of the throat also have been observed. So, what is the pathogenesis of bubonic plague? You can see the flea, it feeds on the infected rodent, and when they ingest the blood within the flea body, the Yersinia pestis organism inside the blood start producing coagulae, so blood turns into a clot. So this flea can be called blocked flea, because food cannot enter through the preventriculus of the flea, and flea feels hungry, start biting everyone. It doesn't matter, animals or humans. Then the aspirated contaminated blood is regurgitated into the biting site. If it bites humans or animals, doesn't matter. And usually the place where we see where the flea bite, so we can see their ulceration, and the initial infections occurs locally in the primary site of entry. So you can see here a picture, the very old picture from the Middle Ages, from the 14th century, where you see that doctors wearing a very special costumes and masks, they examine the body of the human being died of plague, and you can see the buboes or inflamed lymph nodes. So, when bacteria are inoculated through the biting site, so it starts multiplying and cause local inflammation. Then phagocytes come up there, multiply, or engulf these bacteria, and bacteria multiply in monocytes. So, within the incubation period, the organism will be taken to the lymph nodes. And usually the inguinal lymph nodes are involved and start to be inflamed. And that's why the disease had been called bubonic, because bubo means growing. So hemorrhagic inflammation occurs in the lymph nodes and necrosis, and it develops already. Sorry. Ой, Ксения Автандиловна, у меня лекция, можно я вам перезвоню? Хорошо, я через часок перезвоню, спасибо. Dissemination via the bloodstream leads to the sepsis, with development of gangrene of the skin, fingers, penis, meningitis, pneumonia, pleurocarditis, and any other very dangerous state that may lead to death. So, usually the plague occurs in a cycle. That's words about the epidemiology of it. So, we distinguish two types of cycles, wild cycle and domestic cycle, or they also can be called sylvatic cycle and urban cycle. So, sylvatic cycle occurs within the wild animals, wild rodents, but also carnivores may be included into this cycle if flea bites carnivores. And then, without any humans here, the Yersinia pestis may circulate there. And also, the cycle may occur within the domestic rodents and also domestic carnivores, by the way. And through this cycle, it also may enter by biting of the infected flea. So then, if this disease occurs in human as a result of the dissemination of the disease through the bloodstream, the Yersinia pestis may be taken to the lungs. So, as a result of initially bubonic plague, the secondary pneumonic plague can be developed. Why secondary? Because it migrates from the initial site of entry, and later on, as a result of dissemination, it causes an infection in the lungs. But then as soon as the pneumonic plague occurs, then the infected person starts to be able to spread bacteria through the air droplets. So when we have a case of pneumonic plague, so it means that plague will be able to be transmitted from human to human directly. So the pneumonic plague may occur within the human population with no regard to any animal. So like in a third cycle. So that is the very well-known epidemiology of the Yersinia pestis organism and disease that it causes. So how we are going to diagnose this very severe disease? First, we're supposed to collect the sample, and it can be the image smear of the inflamed bubo taken from the patient, or lymph node taken from the patient. And you can see here the image smear stained by Loeffler staining, metal and blue staining, where you can see the safety pins appearance. This is the microbe that has exhibit the bipolar staining here, right? So, and they may be stained by metal and blue, Giemsa staining, but also they can be stained by immunofluorescent stain. So, direct immunofluorescent assay is usually used to demonstrate the presence of Yersinia pestis organism. But in any case, even if you demonstrate it, we have to perform culture. All cultures must be handled with extreme precautions or caution, because it is extremely dangerous. Materials are cultured usually on makonki agar or blood agar. We can use serological tests to identify antibodies against Yersinia pestis. Usually, complement fixation test is used to find the antigen in the infected tissues to find the thermoprecipitated antigen. Usually it is used for examination of putrefied tissues, and then we have to demonstrate the contagious of the disease for the animals. So guinea pigs inoculation also can be used. So nowadays the treatment of plague may dramatically decrease the mortality rate of the disease. So, and if treated, the mortality rate of bubonic plague dropped to 5%, but then the mortality rate of pneumonic plague still can be up to 30%, and any delay in treatment of this disease, especially pneumonic plague, may lead to death. So, all the good antibiotics can be used for treatment, as usually happen with the nosocomial, the agent that cause nosocomial infections. Nobody really treated animals with antibiotics. So that's why Yersinia pestis don't have this antibiotic evolutionary press. So that's why it patients respond very good for the treatment of streptomycin, tetracycline and others. And the drug resistance is not naturally formed in Yersinia pestis. But of course it's better to prevent the disease, and we may distinguish general prevention or prophylaxis that is performed not against this particular microbe, but against a group of microbes that, for example, can be transmitted with the fleas from the rodents. For example, general measures to prevent this disease is the control of fleas and rodents in domestic area, but also in the sylvatic area. Then specific protection can be done by vaccination only, but because the disease is very severe, the vaccines are not very effective. So we may distinguish at least two. A formalin-treated whole-kill vaccine that you can see here is recommended as pre-exposure vaccine, because for high-risk groups, animal handlers, laboratory workers, then foresters, rangers, and travelers to endemic areas. So, and the second type is live plague vaccine, and this is the Russian vaccine from the non-pathogenic, not highly pathogenic so-called EV strain that is given usually per os, and both of these vaccines can prevent bubonic plague within year to 2 years, but then the idea is that they cannot prevent the pneumonic plague. So that's why now we finish with the Yersinia pestis and go for unusual group of rickettsia that we usually don't study, and you're supposed to study them and as independent students work. But then we look through it very briefly to let you know at least something about this group and the most dangerous microbe from this group. So this group had been rearranged regarding taxonomy and classification many times. So, and still the classification is not really established very well. The thing is that these organisms are located somewhere in between bacteria and viruses. Altogether we have several groups there. They are Rickettsia, Orientia, Ehrlichia and Coxiella. and they are aerobic gram-negative bacteria, and the shape is cocobacilli like this, that are obligate intracellular parasites. Can you pay attention to it, please? They cannot develop themselves outside the human cell. So they are obligate intracellular parasites, and the transmission occurs through the arthropod vector as part of their natural cycle. So what can we say about the structure and physiology of these unusual bacteria? If we are talking about structure, they may have cell wall, but even this is a quite typical structure for gram-negative bacteria. They may have very thin peptidoglycan layer. They may have lipopolysaccharide, which has very weak endotoxic activity. They also may have polysaccharide slim layer as an analog of the microcapsule. They also may have some fimbria and pili as the tools for conjugation, and they don't have flagella, so they cannot move. Then they are parasites of eukaryotic cells. They are very small, 0.3 to 2 micrometers. And usually we don't use gram staining for them because they are not stained properly by gram staining. Then how about the culture? So because rickettsia are intracellular parasites, and because they are really very hazardous, very few laboratories can perform their isolation. Usually it is not performed. But then, how can we grow them? Not in a typical bacteriological culture medium. They will not grow in a culture medium. So usually they are isolated from the blood clot, and it can be inoculated into guinea pigs or some animals, tissue cultures, or embryonated eggs into yolk sac. So they are cultured exactly like we culture viruses. Culture technique is very difficult, and these bacteria are placed into biosafety level 3. What diseases are caused by rickettsia? So quite many of them are medically important, so they can be placed into several groups. So at least 2 are given here: typhus group and spotted fever group. By the way, the spotted fevers were also considered, had been considered once as the very dangerous diseases. So typhus group: Rickettsia prowazekii, that causes epidemic typhus and is louse borne, and it is the only anthroponotic disease because it occurs in humans only. And we also may have Rickettsia typhi in Latin, that may cause endemic typhus. And this is zoonotic infection, and humans can be just occasional hosts for this bacteria. The second group is represented by Rickettsia rickettsii, Rocky Mountain spotted fever, Rickettsia acari, rickettsial pox as a disease, Rickettsia conori, Boutonniere disease. So usually they are transmitted through the biting of the ticks or mouse mite. These are soft ticks. So, and usually rodents are hosts for them, or house mice. So this is zoonotic infection. How the infection occurs? The thing is that to cause infection, rickettsia need to enter the cell. It will be usually phagocytosed by monocytes or macrophages, and usually rickettsia may lyse the vacuole that is formed inside the phagosome, and they enter the cytoplasm and can multiply there. The escape from vacuole is mediated by enzyme produced by this bacteria that is called phospholipase. Rickettsia multiply in the host cell cytosol by binary fission like any other bacteria, and then depending on what type of rickettsia do we discuss, they may get out of the cell in different ways. But then anyhow they cause death or damage to these cells. And while they damage infected cells directly, the most common clinical effect of it will be vasculitis and the appearance of the rash on the body. So that's why they may be called spotted fever also. So they can move from cell to cell by mobilization of actin-myosin interaction and propelling themselves from one cell to another one. So pathogenesis of the diseases depend on how rickettsia get out of the cell. So, and here you can see attachment, phagocytosis, escape into the cytoplasm. So multiplication of rickettsia by binary fission, and there you can see the way how rickettsia may get out of the cell. So rickettsia prowazekii rupture the cell, so they are most dangerous rickettsia. Rickettsia typhus may form the core focal lysis of the cell. That's also dangerous for the eukaryotic cell, may cause the death of it. And some of rickettsia can cause least harmful effects on the cell because they may be released by exocytosis, so the cell could survive for very, very long while. So, brief history of this disease, epidemic typhus. So, the records of the cases, or this very good descriptions of the cases, is of year 1083 from a convent near Salerno, Italy. The most severe epidemics of epidemic typhus usually accompany with the wars, natural disasters. So one of the biggest epidemics occurred during the Napoleon's retreated from Moscow 1812, when more French soldiers died of typhus than from Russian bullets. One could say that lice have defeated Napoleon's army. Moreover, the retreat was spread the infected lice and typhus through the entire continent of the Europe and to France directly. During and immediately after the World War I, 30 million cases occurred within 3 million deaths. That's quite a lot. So epidemic typhus is caused by Rickettsia prowazekii, named after von Provacek, the researcher of the epidemic typhus that died of epidemic typhus. What is the duration of incubation period? About 14 days. Clinical manifestation may be seen as fever, severe headache, abdominal pain, chills, rash is very important symptom of typhus together with the fever. But then myalgia, arthralgia, central nervous system involvement with delirium and seizure may be seen during the disease. It is quite fatal. This disease is quite fatal from 10 to 30 of cases. Brill-Zinsser disease occurs, and it is called Brill-Zinsser as a recrudescence of the previous infection with Rickettsia prowazekii, because some of Rickettsia may enter in the human body the unculturable state, may stay dormant for decades, for example 20 to 30 years, and then they may cause disease 30 years after the initial primary infection. But then the recrudescence disease is usually milder than the primary epidemic typhus, of course. How do we diagnose epidemic typhus? So microscopy is quite important. We may take a specimen and perform immunofluorescent test and Giemsa staining, and you can see as a result of Giemsa staining Rickettsia that is stained dark purple, and the rest will be seen blue. Immunofluorescent test exists to detect IgM antibodies. These are the first antibodies produced against these bacteria appear in the blood. And indirect hem agglutination assay may be performed on the lattice particles, and also some of today's very common ELISA Western blot tests. During the World War II, the whale Felix test was really very popular because it was very cheap, simple, and it was based on the cross-reactivity of the antigens of Rickettsia with Proteus vulgaris, 1 more gram-negative bacteria. So antibodies against Rickettsia could also react with the Proteus vulgaris in the agglutination test. So that was really very simple. Culture is not really used for routine diagnosis because it is extremely dangerous to culture this bacteria. Treatment and prevention. Again, because it is zoonotic infection, treatment can be performed with old good antibiotics like tetracycline and chloramphenicol. Whole phenol-killed vaccine is available, and also attenuated live Rickettsia prowazekii vaccine against epidemic typhus also can be used for treatment. Okay. So, then, what happens? So, так. Hold on, I will. Дима, что-то я тут клацнула. Водила, и куда-то я это все увела.
Вот.
Где-то вот это?
Не, вот презентация.
Ага. Вроде я понял. Так,
так.
вы хотите сказать, что у меня только один человек присутствует?
Ну, это будет запись. А в любом случае запись останется, и они просмотрят ее.
Ага, хорошо.
Ну, не то.
Ой, не то. Вот это. Черт.
То есть, в принципе, ничего страшного в этом нет.
Да? Хорошо. Давайте, все, спасибо. Тогда я продолжу. Okay, so that was some technical fixing. So now, I would like to describe one more microbe that can cause really very dangerous disease. And the microbe's name is Borrelia recurrentis from spirochetes. Do you remember that was a group of spiral-shaped bacteria with unusual shape? So Borrelia recurrentis is a causative agent of louse-borne epidemic, relapse and fever. So out of other Borrelia, we pay attention at this one because that's this is anthropootic disease. You can see that the only humans can be the hosts of this organism. And it is also transmitted through the louse. So, pediculus humanus, that's what you studied in the biology department. Few words about the morphology and structure of the spirochetes. You can see the picture, and these are gram-negative microaerophilic fastidious helical rods. So this is scanning electron photograph of it. So the helical rods, very long, up to 30 micrometers in length, and very, very thin, up to 0.5 micrometers in width. So with 3 to 10 loose spirals. So, and this is a blood parasite. It occurs in the blood of infected animals, and it is not usually stained by gram staining because it will be almost invisible. So to make it visible, we usually use giemsa staining. The helical shape of Borrelia is imparted by the periplasmic flagella. That's what we have, periplasmic flagella. We can see cross-section, and in a cross-section you can view this the periplasmic flagella. Flagella insertion points are located near the terminals of the both poles of the spirochetes. Bundles of flagella wind around the flexible rod-shaped protoplasmic cylinder, around this cylinder, and overlapped in the middle. So they meet together in the middle, going from here and from here towards the center of the organism. And the outer membrane constrain the flagella bundles within the periplasm. So it is covered from outside with the outer membrane. So you should understand that flagella don't protrude to outside. So why do we call this disease as relapsing fever? After a person is exposed to infected arthropod, Borrelia spread in the bloodstream, and it spreads to multiple organs, followed by a massive spirochetemia. So, and spirochetes also invade through the endothelium. So, when we have spirochetemia, we can see the first febrile so-called episode. So, because during this episode, Borrelia enter the bloodstream, multiply in the bloodstream, and due to the autolysis, the endotoxin, because this is gram-negative bacteria, may enter into the bloodstream, and this first episode can be really fatal. So if the patient survives this first febrile episode, then the afebrile episode happens. Why it happens? Because within the first episode the antibodies are produced, and antibodies will eliminate the spirochetes from the blood, but not from the body. So that's why we have it for a few days. But then the very important property of these rickettsia to undergo antigenic variations within this period. So within this period they undergo antigenic variations and escape the antibodies that was produced against the initial antigenic type. Then they multiply within this afebrile episode, and then they are released into the bloodstream again. So that we have the second relapse or second febrile episode. And usually it is shorter and is not that severe. So every episode of spirochetemia is terminated as a result of development specific antibodies. You see the specific antibodies are produced here to eliminate the rickettsia, then new antibodies are produced here, then new antibodies, IgM antibodies are produced here to eliminate the microbe from the blood. Then for how long it may last, for how many episodes it can last. So usually around 10 to 13 episodes may occur, but then it will be terminated when because bacteria cannot change the antigenic structure forever. It has limited number of the genes rearrangement, so sooner or later it will revert to original antigenic structure. So then, by that moment, the patient would have all antibodies against all antigenic variants of Borrelia in the body. So then, even without treatment, it will be over. So usually the initial episodes are longer and the later episodes are shorter. So the febrile episode terminates in a crisis with an elevation of the temperature, pulse and blood pressure, followed by a fall in temperature and blood pressure, and they may last for several hours. You can see here that every time the febrile episodes start to be lower, not so severe and not so severe. And the disease can be severe and death occur in 10% in 10 to 40% of symptomatic cases in the absence of the treatment, and in 2 to 5% still, but that is still high mortality rate in the persons that are treated with the antibiotics. Laos-borne relapsing fever, this is abbreviation, is endemic only in Northeast Africa right now. These are Ethiopia, Sudan, and some other countries. And was recently diagnosed in Europe when a lot of refugees from these countries arranged the camps in these European countries. So it tends to occur in epidemics, mainly in the regions affected by war, refugee camps, due to some disasters like floods and so on. And you can see here the soldiers in World War I that are searching for lice, because that's how they could prevent the transmission of this Borrelia. So, if the louse is crushed on a new host, Borrelia are released and can enter braided skin or bites. Louse leaves the host within fever episode and infest a new host with normal body temperature. So, louse or lice, they don't like the humans with high temperature that doesn't fit with their physiological features. How do we perform diagnosis of louse-borne relapsing fever? You should understand that if we have the microbe that may undergo antigenic variations, the serological tests, as you see here, are really unreliable. So we never know which kind of antigen we're supposed to find. So the specimen should be taken during the febrile episode, because within this febrile episode we have highest concentration of rickettsia in the patient's blood. And then it is confirmed by visualizing of the spirochetes in this blood during the febrile episode. So how do we see them? So first, dark-field microscopy can be performed, or staining. The most common is Giemsa staining of the thick and thin blood smears. Yeah, that is the most common one. Most patients with this disease are diagnosed just by microscopy, and that will be enough, because we could see directly the organisms in the bloodstream. Now the molecular diagnostic tests to diagnose the relapsing fever are developed. So they are available, but can aid in diagnosis of relapsing fever. So how about treatment? It responds quite good for antibiotic treatment with the tetracycline, penicillin G, erythromycin, or some others. So the prevention of louse-borne relapsing fever relies on measures to avoid infestation of the body with the flea. Vaccines are not available against this disease, so that's why it can be recognized as potential warfare agent. Then we switch to Vibrio cholera. I will give very short description because one more left, that is Bacillus anthracis. Vibrio cholera, and you can see the scanning electron microphotograph of the microorganism, and that's how we see it under the compound light microscope. So cholera, they cause cholera, is a potential epidemic and life-threatening secretory diarrhea characterized by numerous watery stool accompanied with vomiting that may lead to hypovolemic shock and acidosis and death. So Vibrio cholera is a very small 2 to 4 micrometers in length, gram-negative bacteria, curved, right, with one polar flagellum, highly motile. This one flagellum gives very high motility to the organism. It is aerobic, but mainly facultative anaerobes. So others, but Vibrio cholera itself is aerobic bacteria, and it has very high fermentative properties, really very pronounced properties. It grows very well at the wide range of temperature from 18 to 37. It means that occasionally it may grow in water, in fresh or brackish waters. So they also are known for their very high pH requirements, up to 9.5. And this property of Vibrio cholera can be exploited in the selective isolation of these bacteria from the specimen, stool, or from environmental samples like waters. Vibrio cholerae is very susceptible to heating, drying, and acidity of the gastric juice, but it is really very stable in sterile seawater, fresh water, and it may survive for a month in even in tap water treated with chlorination. So the classification of Vibrio cholera organisms is really a bit tricky. So first, it is based on the antigenic structure. So if we take a species, Vibrio cholera, it can be classified based on antigenic structure into 139 serogroups. You can see it here, right? You don't see any in between. So because those that are in between O1 and O139 are called non-agglutinable Vibrio cholera organisms. So of course they will agglutinate with the serum against O2, or for example O3 with O3, but then they are called not agglutinable because they will not be agglutinated with O1 serum and O139 serum. So then out of all 139 serogroups, the only O1 group and O139 group cause true cholera. But then regarding further classification, O1 serogroup can be further classified due to the minor antigens of the cell wall into 3 serotypes. So GABA, INABA, Hikojima, these are the names of Chinese microbiology that perform this. And these all serotypes that can cause Vibrio cholera disease, true cholera. O139 is not further diagnosed into serotypes. So, apart from this is antigenic classification based on antigenic structure. But O1 serogroup Vibrio cholera can be classified based on their biological properties. So what does it mean, biological properties? Some fermentative properties, the phage typing, so ability to be infected with the particular specific bacteriophages, and ability to produce hemolysis. So Vibrio cholera organisms, O1 organisms, can be classified into 2 biotypes: classical biotype and L-Tor. That's it. So, what is cholera as a disease? So you know, the way of transmission is fecal-oral through contaminated food, but most commonly water. So the gastric acidity provides an effective barrier. So people more susceptible with achlorhydria or hypochlorhydria, and the infective dose can be reduced from 10 power 3 to 10 power 5 organisms. In the small intestinal tract, Vibrio cross the protective layer here and reach epithelial cells with the help of high motility. We mentioned that one single flagella gives very high motility. Then chemotaxis, adhesive pili, and the ability to produce mucinase to dissolve the mucin. And then there, cholera may produce the cholera exotoxin that is called choleragen. Why it is called so? Because this toxin generates cholera. It is responsible for cholera as a disease. How does it work? You can see quite nice picture. Here you can see that this is a cholera toxin together. And it binds with the GMI1 receptor with the help of B structures, and altogether we have B submolecules, and B means binding. Then they enter the cell through endocytosis, and they are transported to Golgi apparatus, and subunit are cleaved there, subunit A into 2. Then the subunit A is transported into the Golgi, and A subunit interact with G protein. G protein target adenylyl cyclase activation, and adenylyl cyclase converge ATP to CAMP and cause accumulation of CAMP, and it is in turn cause hypersecretion of water and electrolytes from the cell into the lumen of the gut. That is the origin of difficult to stop diarrhea. So, and diarrhea is the major symptom, as we mentioned. It is responsible for loss of fluid, and if not treated, so it is responsible for hypovolemic shock. So how do we perform cholera diagnosis? So, by the way, diagnostic procedure is quite difficult because it has very high biochemical activity, so that's why many biochemical tests are supposed to be performed. But then, for diagnostic approaches, the classical approach is culture. So as soon as we get culture, and usually we culture it on the alkaline broth and alkaline agar. Why? Because this is a selective medium to get rid of contaminating bacteria, because they won't grow at so high pH up to 9.6. As soon as we get culture, then we perform biochemical tests for identification, serological tests for identification whether it is O1 or O139, biotyping to know if it is O1 whether it is classical or L-TOR biovar, by phage typing and some other tests. But frankly speaking, now the molecular diagnosis is performed very often. And within the molecular diagnosis, you see here PCR. In one single amplification round, in one single sample, we may simultaneously check the presence of genes for O1 lipopolysaccharide, so because that's the only one. Then we may also check the presence of the gene that code for toxin coagulated pillars, so-called. The thing is that only those Vibrio cholera organisms that have these pili are able to produce cholera toxin, because it also in turn encoded by bacteriophage. And then cholera toxin. So we're supposed to have primers for 3 antigens: O1 antigen, TCP pilus antigen, and cholera toxin antigen, genes for those antigens. So that's why we can have 3 pairs of primers, and in one single PCR we may identify whether it is O1, whether it's toxigenic or not, and whether it's epidemiologically important. It means if it has TCP pilus and can be toxigenic or cannot be toxigenic at all. Treatment and prevention of cholera. Cholera can be life-threatening disease, but it can be easily treated. And of course, the most important is immediate rehydration, replacement on fluid electrolytes. And antibiotics are used for treatment also to shorten the duration of the illness and the severity of the disease. But rehydration therapy is more important than antibiotic therapy. By the way, for prevention of this disease, several vaccines had been developed, and they also can be recommended for travelers to the high-risk countries. But it may not be protective against all strains of Vibrio cholera, and the protection cannot last long. Not so far ago, a comparatively new type of vaccine had been developed, that is conjugative vaccine, where the toxoid or non-toxic portion of the choleragen toxin is conjugated with O1 lipopolysaccharide antigen. This vaccine by now is considered as the best, but still the duration of protection is not so high. And now I would like to end with the Bacillus anthracis as a causative agent of anthrax. And you can see here the very old picture where Robert Koch working in his lab, because he is the one who discovered Vibrio cholera organism. Bacillus anthracis organism. But he also worked with Vibrio cholera. So, this is pathogenic bacillus that causes anthrax, an important disease of animals. So this is a zoonotic disease, but occasionally it may be transmitted to humans, and it could be a major agent of biological warfare. So in this genus we may have some other bacteria, but of course we are interested in Bacillus anthracis. This is a quite large rod, gram-positive, you can see it here, and it is with square ends, and it gives the appearance of bamboo stick in the slides. It forms long chains, so they are arranged in chains, and you can see like holes or opens in the organism. So this is the process of sporulation occurs. So the spores can be formed, and usually they are formed centrally, but subterminally or terminally is also possible. So they are motile. Other bacilli, except for Bacillus anthracis, it is not motile. It is mesophilic, and thermophilic species there could occur, but then the Bacillus anthracis itself is mesophilic. So, and of course, Bacillus anthracis is the one of medical importance. It was the first bacilli isolated in pure culture and shown to process spores by Robert Koch. And then that was the first bacterium used for preparation of the live attenuated vaccine by Louis Pasteur in 1881. So this is the organism itself. And in tissues, if it is found singly, it usually surrounds by the capsule. And the end of the bacilli may be truncated and swollen, so it gives the appearance of bamboo stick. So the capsule can be stained. There are several ways to stain the capsule. Because the capsule is polypeptide nature, it can be seen due to the McFadden reaction, and it will be purplish, and the organism itself will be blue. So it can be stained also by negative staining, then the capsule will be non-stained, or by immunofluorescent assay, you can see with fits. So how it appears on media? Because pathogenic Bacillus anthracis organisms may form capsule, The colonies of encapsulated bacteria will be mucoid on mid-peptone agar. So, under ordinary conditions, the capsule is formed or in the human or animal body only, but not outside in the laboratory. So, unless we add some special substances to force bacteria to demonstrate capsule, to form capsule. That could be bicarbonates or serum albumin or starch. So spores usually are central and they are elliptical. Few words about the spore. Do you remember when we studied them in general microbiology? We mentioned that they are highly resistant to drying, heating. They stand the 2 hours boiling. So why it is so? Because there is a special structures that cover spore. Take a look here. First, this is exosporium. Then the second is spore coat. The third is cortex, very thick layer. The next one is core wall. There's the remains of the bacilli cell wall. Then inside the spore we have nucleoid. This is a genetic material, and very few ribosomes to start multiplication as soon as the spore will be placed into viable good conditions. So, how do we culture Bacillus anthracis? They are aerobes, Bacillus anthracis itself, and it can be culturable within the temperature from 12 to 45 degrees Celsius. And in broth, it causes formation of floccular deposit with little or no turbidity within the medium, and it may grow on ordinary medium, and the colonies will appear as cut glass, but they also grow on the blood agar. So the colonies of Bacillus anthracis, because see, they grow by dividing and by elongation of the chain, they adjust to, so they may form the colonies of the medusa head appearance, so is covered by matted hair. So sometimes we need to distinguish Bacillus anthracis with many other bacilli, non-pathogenic bacilli. So it is non-hemolytic. Why? For example, Bacillus cereus is hemolytic and may cause visible hemolysis on blood agar. Bacillus anthracis is capable of producing proteolytic enzymes, so it possesses proteolytic properties. If we perform inoculation or puncture of the gelatin layer, so after 2 days of incubation, bacteria start growing, producing proteolytic enzyme and dissolving gelatin. So, and gelatin liquefaction area may have the appearance of inverted EV tree, like here. Then the very important is diagnostic reaction is strain of pearl reaction. You know that they are rods, but then if you culture Bacillus anthracis on the solid medium with the addition of penicillin, not so much, within 3 to 6 hours, then you can see that the organism turns coccus, start to be spherical, enlarged, but still occurred in chain, like a string of pearl or bracelet or whatever. So the reaction is used to differentiate with other bacilli. So usually for selective isolation, the very special media is used, and this is mainly for isolation of bacilli from the animal corpses. So the Platt medium is used. Platt, P stands for polymyxin, L-lysozyme, E-ethylene diamine EDTA, and T-tellus acetate added to heart infusion agar. And that's for getting rid of other spore-bearing bacilli, not Bacillus anthracis. So it may demonstrate some biochemical reactions. For example, carbohydrate fermentation can be performed with acid formation but no gas. So nitrates can be reduced and catalases formed, but they are not really very often used in identification of Bacillus anthracis. So what makes it very dangerous? Of course, the vegetative cells are not very resistant and can be destroyed easily by boiling or heating up to 60 degrees Celsius within 30 minutes. But the spores of it are highly resistant to environment and may persist for years in dry earth. So by now it had been measured for 60 years, and for 60 years the spores of Bacillus anthracis stayed viable. The spores can withstand dry heat and certain chemical disinfectants. For example, it survived in phenol for several weeks. So that's why this sterilization procedure is very important of the product that can be contaminated with the spores of Bacillus anthracis. And these are either skins of animals, hair, wool, bones, carcasses of the animals. And all these things should be sterilized by autoclaving. What do we know about the disease? Generally, it is a disease of herbivores, of grazing animals. Other animals, like for example small rodents, are resistant to this infection. So humans can be just accidental hosts of this disease. They be contaminated, they be infected by the contact with dead infected animals or their products. Usually contamination or infection occurs through the spores, and spores cannot be found on a live animal, just in dead animal. Bacillus anthracis is one of the most serious biological warfare agent. We stated that there are natural sources of this infection in environment. So you can see here how many different types of animals may be a source of infection. What are the virulence factors of Bacillus anthracis? First, we paid attention at the proteinaceous capsule. It is highly phagocytic. So Bacillus anthracis can produce exotoxin that is made of 3 proteins, and we can see here how these proteins act. See, this is a Bacillus anthracis that release 3 protein molecules. One is called protective antigen, second one is edema factor and lethal factor, second and third. So how it occurs? When these molecules are released, protective antigen binds with the anthrax receptor, or Bacillus anthracis receptor, and will be polymerized. See, so many molecules may combine together. So with many anthrax receptors. And then on top of this structure, either lethal antigen or edema antigen can be fixed. And then endocytosis occur, and this structure, protective antigen together with the active molecule, will be incorporated into the endosome. And as a result of this, from the endosome, lethal factor and edema factor will be released, and edema factor causes the release of fluid into the intracellular space, and lethal factor as metalloprotease causes death of the cell. Okay, my dears, I think I need to finish because the next lecture is coming, and the anthrax may be seen in several forms. The cutaneous anthrax is not so highly dangerous. The lethality can be up to 5%. Gastrointestinal is very rare, so the inhalation anthrax is the most dangerous form of anthrax. See you guys in the classes, and the next week lecture will be in immunology. Goodbye, my dears. See you on classes. Все, Дима.